Good morning, everyone. Nice to see so many people. My name is Ricarda Kater, or too many of you I'll be mainly known as Ricky, because it's easier to remember. I've been the PhD student working at the University of Sheffield on the EARTH project, which is sponsored by the East Anglia beekeepers, together with one of the research councils. And my research has focused on how the Varroa mite stays hidden inside the hive and how it takes advantage of the way that our bees naturally are able to identify each other. So usually my research investigates how your bees can tell each other apart and how they can recognize strangers inside the nest. And as part of this project, I've been focusing my studies on how the Varroa mite interferes with this recognition system. So what I thought, I'll give you an introduction to the Varroa mite, because I still come across quite a lot of beekeepers who've never seen the mite and don't quite know what it does and how it actually ends up in your hive. And then I move on to the research itself. So can I just see how many people of you have ever actually seen the mite? You know, with it. Excellent. That is good to hear, because I still get quite scared when people have not heard of it, and I'm like, you know, it'd be better to know what it looks like, you know, if you're looking for it in your own hive. So, as you all know in that case, the biggest problem with Varroa is that it can transmit viruses such as the deformed wing virus, and the typical symptoms are, you know, deformed wings as well as paralyzed behavior in young bees. And it's quite interesting because when the mite feeds on adult bees and transmits the virus, these symptoms don't occur. But if the mite feeds on larvae or pupae, it really interferes with the natural development of the bee, and that's when you get these sort of glued together wings. And these bees, I mean, you can tell already just by looking at them, they don't last very long. And if you have a lot of these in your hive, the colony will collapse very quickly. And the problem with that is that you can actually go into winter with a very strong colony, but if you have mites that carry this particular virus, you know, you can sort of feed your bees as much as you want, they won't survive winter. And this is a particularly big problem in America, as well as, you know, in Europe, whereas the Asian honeybee doesn't have this sort of problem because they have a different way of dealing with Varroa. But unfortunately in Europe and America and Canada, this is still a very big problem. And also causes a lot of damage, economic damage, so actually very costly, especially if you have, like a lot of American beekeepers, you have hundreds of thousands of colonies, and half your, you know, apiary dies of that. You know, it's very costly to keep this disease. The Varroa actually, as it says, you know, is a Varroa mite, so it belongs to the mite family, which also contains the red spider mites and dust mites. So it's not an insect, but it's, you know, it belongs to the acaricides. And it's actually not that uncommon for mites to parasitize insects like bees. So it's not only the Varroa mite you can find on honeybees, but you also get solitary bees that can carry, for example, this sort of mite species that lives as a sort of colony on top of the solitary bee. You find bumblebees regularly that have tiny little mites living in amongst their hair. And sometimes they feed on the bee's blood, on the bee's hemolymph, and sometimes they just feed sort of from leftovers, you know, pollen or nectar that the bee carries around. And even beetles can have mites. And you can see here, if the infestation is quite badly, how, you know, I mean, this must be a lot of weight carried, you know, to be carried around if you're a beetle. And the mites will actually lay eggs all along the insect and live on it. And even the smallest of insects, such as, you know, tiny ants, you get this problem. And if you do research on ants in the lab and you have a mite outbreak, there's actually not an awful lot you can do, and you have to get rid of the whole colony. So it's quite a big problem to researchers as well. And it's not just confined to bees. And even mites can contract other mite parasites. So they're not immune to it. You'll be happy to hear that. But unfortunately, there's no sort of mite that feeds on the Varroa mite. But, you know, they can contract mites themselves. So moving on, or going back to Varroa, how can your hive actually end up with the Varroa infestation? The way that Varroa moves around is it stays on the bee body most of the time, and it just switches hosts, it switches bees depending on where it wants to get to. So if bees accidentally end up in your hive, bees from another infested colony, which could be a wild colony or someone, let's say your neighbor's hive, and they accidentally drift into your hive or they try and rob it, this is the main way how Varroa actually ends up in your hive. And when that bee is then inspected by the guard bees of this particular hive, just this, you've probably seen this before when they antennate each other, they're getting quite close, and this is the point where Varroa will actually run from one bee onto the other bee, and they're very fast Varroa mites. So this happens in a split second, and what the mite then tries to do is when the guard bee then comes across another forager, it moves on to that. If the forager then comes across another nurse inside this hive, the mite moves on to the nurse. And the nurses are really the ones that the mite wants to get on once it's inside the hive, because nurses look after the brood, and that's where the mite wants to reproduce. So your best strategy if you are a Varroa mite and you are sitting on a forager bee is to then switch onto a nurse bee as soon as that contact has been established. When the nurse bee, for example, has been begging for food, forager is feeding her, that's when the mite moves across. And it basically then sits on the nurse bee. So you can see that here when they antennate each other, it's quite easy for the mites to move from one bee to the next. If the nurse bee then is in the brood area and attends different larvae and pupae in the brood cells, the mite sits on the bee, and again it waits for the nurse bee to come across a cell that is just about to be capped, and all of this is being done using odour. So the mite can distinguish between forager and nurse bees based on odour, because you have to remember inside the hive it's dark, so you can't use your eyesight, and mite's eyesight is not very good anyway, the mite can even identify brood cells of different ages just based on the odour that the brood emits. And this is the main way how the Varroa mite actually finds its way around the hive. When it comes across a cell like that, it'll then drop off the nurse into the cell and hides in the brood food at the bottom of at the bottom of the cell. And it even has a special snorkel that helps it to basically swim in the brood food and still be able to breathe while it's doing that. It waits for the cell to be capped and then feeds on the pupa after the cell has been capped and starts laying its eggs. The first egg is always laid at the entrance of the cell, the other 4 eggs at the bottom. The first egg at the entrance is always a male, and there's only 1 male per mite family. All the other eggs are females. The male emerges first and then tries to find the females inside the cell. Usually it waits for them to emerge, and then the mating happens inside the cell and it's purely inbred. So if you have 1 mite family inside the cell, the mating happens within that family. Sometimes when the mite infestation in the hive is very high, you can have 2 families, but usually it's an inbred system. The mother mite doesn't care a lot for the young. What it does do, though, is it creates a little hole in the pupa that makes the access to the blood easier for the young. So there is a little bit of sort of parental care going on, but that's sort of the beginning and the end of it. The mite larvae and immature stages will then feed on the pupa and develop inside the cell. And this is typically what a mite family would look like. So you have the male on the right. And you can sort of see, you can see that the male is definitely considerably smaller than the mother mite and even its sort of older sisters. It also has a more pointed shape and it's slightly green in colour. And the males will never get as dark a colour as the mother mite. And this is also the reason why when the mite family leaves the cell, the male dies, because this brown colour is a clue that the cuticle is quite thick, which protects the mite from losing too much water. So desiccation is a very big problem in mites as well as in insects. And this also means that when the mite family leaves, the whole mite family leaves the cell when the bee eventually emerges, all the mites at these early stages that are still quite white, they will die because they basically lose so much water. Because if the cell, the brood cell is sealed, the humidity inside the cell is quite high, but if you take that seal away, it drops quite dramatically, and if you don't have that thick cuticle, you basically dry out. So your females will usually start from the egg stage into these sort of young, immature stages, and then grow slightly, and after a while develop this dark colour, like I said, which is a sign that the cuticle has thickened. So if you open an infested cell, this is typically what you would encounter, the different members of the family, or if you pull out a drone or a worker bee. The interesting thing about Varroa is that it's actually quite a big parasite if you compare it to its host. So if I'm the size of a bee, Varroa would be about this big. And that raises the questions why the bees don't actively try and push the mites off or attack them, because even if you have them in an observation hive inside the lab, you never really see them going for the mite. And given the size of the parasite, I mean, if I had that big a parasite on me, I would, even if I couldn't see it, I would know that there is something moving around, and I would try and get rid of it. Now, the reason for that, even if the bees try to sort of push it off, or even when they're grooming themselves, the reason why the mite usually makes it quite difficult for the bee to push it off is because it has different adaptations that help it to stay on the bee, such like this crab-shaped carapace. So its body is sort of crab-shaped, and what that allows it to basically push itself against the bee body, and it naturally makes the bee's legs slide off the side. So it's very difficult for the bee, even if the bee grooms itself, to accidentally knock the mite off because it can't hold on to anything. The mite also has special hairs on the side of its body, these ventral hairs, and these serve as sort of hooks to hook the mite into with the bee hair. Again, another way of making sure that it stays on the bee body. And finally, it has special lobed suckers on its feet that again help it to suck its body against the bee. But what was of particular interest to us is that the mite actually uses a chemical strategy to stay hidden from the bee's sort of, recognition system. And in 1995, Nation discovered that the odor that the bee produces bees usually tell each other apart based on the odor that they produce. Like I said, they don't use their eyesight normally, but they use a very complex sort of odor system that tells other bees whether the bee's a nurse or a forager, what age it is, what colony it belongs to. And Nation found that the mite produces or has compounds that are very similar to that of the honeybee. So it seems to mimic the odor of the bee, which is quite clever because you think that the mite then sits on the bee, but it blends in with this odor, and the bee mainly recognizes invaders based on the odor system. It can't tell apart whether a mite is on it or not because there is no difference in odor between the two. Because the main reason how one bee actually identifies another bee is that on the skin of your honeybees, there will be a thin lipid layer. And in this lipid layer are a variety of different compounds. It's almost like different perfume layers. And that's what bees use to tell each other apart. So let's say you have an African honeybee on the left, you have a European honeybee on the right. These are two chromatograms, which basically show you the types of compounds that the bees produce. Every peak is a different compound, and the height of the peak is a concentration. Now, the African honeybee produces more or less the same compounds as the European honeybee, but it produces one particular compound that is absent in the European honeybee. And also the compounds that these two share, they produce in different concentrations. So you see the heights of the peaks are slightly different. So overall, the African honeybee has a very different odor compared to the European honeybee, and this makes it very easy for the two to at least recognize that one is not the same species as the other. So all you need to do if you're a bee is to smell another individual and then compare it to your own odor. And if it doesn't smell anything like it, you know, it's an invader, and you know, you need to react with aggression if that invader is inside the hive, or ignore it if it's outside. You know, it depends on what sort of situation the bee is in. So the mite, like I said, produces the same types of compounds, or has the same types of compounds as the honeybee, which is actually quite a common strategy amongst what we call social parasites. So social parasites are parasites that infest social insects such as wasps and ants and different types of bees. And you have a type of chemical mimicry where, for example, the Uracema wasp, which is a parasite, lives in fire ant colonies. And even though, you know, one is an ant, the other one is a wasp, so they look quite different, and you would have thought that the ant would definitely be able to tell apart, you know, this wasp from other ants in its colony. But because the wasp actually produces the same types of compounds as the ant produces, when it's inside the colony, and again it's dark, so you can only rely on odor, the ants will actually attend to the wasp and feed it, and you know, the wasp also makes little noises and it behaves like one of the ants, so that the wasp can actually go into the brood area and then just feed on the brood completely undisturbed, and like I said, it even begs ant foragers for food and it gets fed. So it's quite a clever strategy to get yourself accepted into the colony and then take advantage of it. Another type of mimicry is what we call chemical insignificance, which is used, for example, by the cuckoo wasp, which invades the nest of the bee wolf. And in this case, the cuckoo wasp doesn't pretend to be a bee wolf, like in the example before, but what it does, it covers itself with a sort of a low concentration of the bee wolf odor. And the reason for that is that the nest of the bee wolf is actually also covered in this odor. And when the cuckoo wasp then enters the nest, it usually stays on the wall, so it will never interact with the bee wolf, but it stays on the walls, and that way it blends in with the odor that's already on the wall and completely disappears, well, for the bee wolf, completely disappears in this sort of chemical environment, which means that it can again make its way undisturbed into the brood area and then feed on the brood and leave without the bee wolf even knowing that it was there. Now, with Varroa, the problem is that actually, as we know, Varroa moves from foragers onto nurses, onto brood, and all of these have quite different odors. So what we found is that when you have a forager individual in your hive and a nurse bee, so one that attends brood, these two individuals here, shown as example, are from the same colony, and you can see just by looking at their profile that they are quite drastically different. And this allows, like I said, foragers to do, you know, identify nurses, and for nurses to identify foragers. But it means that when a mite starts off on a forager, it needs to mimic this odor, and when it then moves on to a nurse bee, it would have to mimic this particular odor. So it's quite a big, you know, difference from here to there. So we wanted to know what Varroa actually does. Does it sort of produce an odor, sort of one that fits all, or does it really adjust its odor mimicry to fit that particular bee, which is quite costly. If you think about that, you would have to, if Varroa actually produces these compounds, it would have to tell its genes to upregulate the production of some compounds, downregulate the production of other compounds. That would take quite a lot of time to do, and is quite, you know, energetically costly. So the first thing we had to find out is, okay, so what does Varroa actually do when it moves from one to another? And we took mites, or we kept mites for 24 hours on adult bees. These are nurse bees. This is the profile of, your average nurse bee. This is the profile that the mite then has itself, and you can see it's very similar. And when we then moved the mites onto larvae, and larvae are quite different in that they have these high concentrated, which are methyl alkanes, for those of you who have a chemical background, which are very low in the adult bees. You can see here the peaks are very low, and then in the brood these shoot up. So when we kept mites on larvae for 24 hours, they went from this profile, which is that mimicking the adult bee, onto a larva profile. So you could see this shoot up in compounds within 24 hours. And again, when we kept them on pupae, so on a sort of slightly later developmental stage, they were perfectly able to imitate that profile and also that of newly emerging bees when we kept them on bees that were just about to emerge. So as the mite moves through the hive, it does indeed adjust its own odour to every single type of bee that it sits on, which is quite a remarkable thing to do, especially when you think about that different colonies also have their own unique odours. So it would have to mimic those as well, and this is indeed what we found. So when we this is a sort of cluster analysis, which means that on the left you have bees that cluster according to their odour. So if they have the same odour, they cluster together. If they don't, it would be sort of a random distribution. And you can see here, based on the colour, that bees of the same colony have very similar profiles, and thus they cluster together. And if you then collected mites from the same colonies, they also cluster according to those colonies, which means that, yes, the mites also produce an odour that fits that particular colony. So not only do they adjust the odour based on the bee, but also based on the colony that they're in. So they're quite highly sophisticated in that sense. So when do mites actually start being able to do that? When we looked at the immature stages of the mite and looked at their chemical profiles, we looked at the male first, and we found that the male, again, right away mimics the odour of the particular larva or pupa that it sits on. And the same was the case with immature females, so really young female mites, and more mature females. And this is quite interesting because usually in insects, males and females produce different odours so that they can find each other. And in the mite system, this doesn't seem to be the case, at least not with the Varroa mite. And the reason for that is probably that it doesn't need to do that, because if you remember that in one brood cell you usually have one family, you only have one male, you typically have 4 females. So the male doesn't really need to know whether there are other males in the brood cell, because it's always likely that the other mite that it encounters is either its sister or its mother. And I'm sure the mother will tell it quite quickly that it's not mating. But unless you get 2 or 3 families inside the brood cell, you don't really, as a male, you don't really need to know whether you're encountering a male or a female. As long as it's a mite, that's fine. And again, they use the sense of touch a lot as well than with recognition in the mites. So they can get around that way. The next thing we wanted to test was, okay, so how quickly can this happen, this sort of change from one profile, from one odour to another? Because if you think about it, your forager ends up in a wrong colony, and then the mite tries to hop from that individual onto another member of the new colony, which means that it will still have the odour of the previous bee. And how quickly can it then produce or get to the new odour? Because within that small, time window where its odour is still different from that on the new bee that it sits on, there is a chance that the bee actually notices that there is something funny on her back and tries to get rid of it. So what we did is we took adult bees and pupae, because these are quite different in the odour profiles, especially in these sort of compounds that I've highlighted in red. So these are quite high in pupae, but low in adult bees. And we took mites from adult bees, they were on them for like 24 hours to make sure that they definitely had that particular profile. And then we moved them on to pupae and left them on the pupa for 20 minutes. And you can already see that from hardly any compounds of these red compounds being there when it's still been on the adult bee, within 20 minutes you can see an increase in the compounds. After an hour they go up even more, and then within 3 hours you can get almost a full mimicry of the pupa profile. So this showed us that when the mite moves from one bee to another, it takes it around 3 hours to then fully mimic the odour of that new bee, which is very fast, given that we know in insects at least, when they just start to produce the odour, it takes them about 6 to 12 hours to really have an odour profile on their skin. So this is the point where we started to think, well, do the mites really produce these compounds, or have they got another way of getting these compounds from the bee without having to produce them, which would be the more sort of cost-efficient, energy-efficient way of doing it? Because other ways to get your host compounds, and obviously the bee in this case is the host, for parasites is to get the compound straight from this sort of lipid layer. And if you remember, I said that bees carry a very thin lipid layer on their skin, which is where their odour sits. It's like an oily sort of layer, which is basically like a perfume layer. And some social parasites take the odour straight from there, whereas others will actually take up compounds whilst feeding on the host. So with honeybees, some of these compounds that are on the skin are actually also in the blood, and the mite could take up these compounds whilst it's feeding on the bee and then basically express them on its own cuticle. Or it could synthesize, so it could produce the compounds itself. So these are the three main possibilities. So what we did is we looked at what mites looked like when they just come off adult bees, which is this left group. The alkene, analkane, and methylalkane are basically the three main chemical classes that make up the odour of a honeybee. And adult bees have a lot of this sort of grey, this alkene, but very little of these, what I showed earlier as red compounds, which are methylalkanes. Whereas pupae have a lot of methylalkane and very little grey, which you can see if you then put the mite, this is the mite profile, onto a pupa, the red compounds shoot up and you get this drop in the grey, in the alkene compounds. However, when we then moved mites from adult bees onto pupae and we had removed this oily layer on the pupa, so the mite was only able to feed on the pupa, and because we kept the bee and the mites in a small vial with holes in them and we placed other pupae around them, they were still able to smell a pupa smell, so they knew that they had to adjust their own smell to pupae. Because the problem is, in this experiment, if you remove the odour of the pupa, the mite doesn't know what it has to adjust itself to. So you still have to provide some sort of odour signal. And we did that by keeping mites and one pupa inside a vial, and the vial had holes. The pupa inside the vial had its odour removed, so we washed it in a special solvent. So they couldn't get this oily layer because we'd removed them, but we then replaced pupae around the vial so that the mite knew, okay, I'm surrounded by pupae, I need to adjust my profile to pupae. But without this oily layer, they couldn't. So you still got this drop in the grey compound, which seems to be a natural sort of wear-off if there's not a lot of alkene around, and obviously pupae don't have a lot of alkene. But they couldn't increase these red compounds, so they needed that oily layer to, get a pupa odour profile, and they weren't able to do that without this oily layer. Then, to check that they could actually mimic a pupa profile with only the oily layer, but not being able to feed on the bee, because we need to make sure that they're definitely not taking any compounds from the blood. So we need to sort of provide evidence that it's only the oily layer that allows them to do that. We took the odour off a honeybee pupa and transferred it onto a pupa of the blue butterfly. And the reason for that is that the blue butterfly pupa has a very thick cuticle, and the mite cannot get through the cuticle and feed on the blood. So there's no way it can get to the blood and then take any compounds up. Obviously, we washed the pupa before we transferred the odour of the honeybee pupa to make sure that it's literally just that odour profile on it. And the mites were quite happy to do it, and we made sure that we fed the mites for 24 hours before, so they weren't deficient of anything. And you can see from the profiles that, you know, this is a profile that we found on the pupa when we spiked it with honeybee pupa odour, so it's the same profile, so we did replicate it in a sort of natural way. And even though the mite only had access to this sort of artificial oily layer on top of the fly pupa, it was able to then increase its own red compounded methylalkane, and again you get this drop in alkene. And it's basically just the oily layer that it needs to get the compound, or this compound class. And then we thought, okay, so is this an active process then, if we're literally just talking about sort of a sponge effect? So somehow the mite being able to suck up the bee odour and then express it on its own, or cover its own skin with it. Because the problem is that the Varroa mite doesn't actually have legs that are long enough to groom itself and transfer all the odour. Because you get, for example, social parasites like shampoo ants, and they're called shampoo ants because they groom the host, the queen mainly, a lot, and then they groom themselves, and that way they transfer odour from the host onto its own skin, its own cuticle. Now the Varroa mite can't do that because its legs are too short. So how on earth can it cover itself with the odour of, you know, of the honeybee? So what we did, we kept mites that were, well, live, so the natural state, on pupae, and then we killed some mites and washed them and put them, and there shouldn't be much sympathy in this room, I don't think, for this, so sorry. I can get away with it. Well, we froze them before we, you know, that's the best way, really, you know, the most humane way of dealing with that. We removed the odour that was on the mites, we washed them basically, and then we placed these dead, washed mites on pupae in the sort of normal way, so with their legs on the body, so not the other way around, and left them on there for 24 hours. And surprise, surprise, you know, they still were able to increase the red compounds, the methylalkane class. So they don't even need to be alive to do this. It's a completely passive process, which indicates that there is something within the mite cuticle, this is sort of the skin physiology of the mite, whether it's all these hairs or whether it produces a special lipid that basically, because lipids are oily, you know, oils love other oils, so they mix naturally. So it could be that Varroa is producing a special oil that basically then sucks up the sort of oily layer from the bee, which is where all the odour sits, and it naturally spreads over the bee body. And this also explains why the mite can so easily sit on, well, move from one bee to another and you know, change its mimicry as it goes along, without actually, you know, it costing it a lot of energy, because it's all happening sort of passively. So just to sort of summarize this sort of chemical strategy that Varroa uses. So let's say we have, you know, a forager from another hive coming into your colony, drifting, robbing, who knows. That forager will have obviously a different odour, shown here in brown, and members of the colony that this forager ended up in accidentally obviously have a different odour, so they're all in yellow. Let's say that forager carries a mite, that mite mimics this forager's odour, and then it comes across, the bee comes across another, or is inspected by another bee from this colony, the mite moves across, and then we know now that within 3 hours, it can adjust its own odour to that of the bee. And that way becomes sort of like a ghost-like creature inside the hive. So the poor bees don't even know what on earth is going on. And during these 3 hours, when it still has a slightly different odour at the beginning, it can make use of all these other adaptations. If you remember at the beginning of the talk, I said, it has this crab-like shape, it has these ventral hairs, it has these lobed suckers. All of these things enable it to actually hold on to the bee until its skin has done what it does best, this sort of chemical this uptake, this spongy uptake of the odour. And you also have to remember that obviously mites love going in between the scales of bees, and that's a very difficult place to get them out. And within 3 hours, they'll be able to then adjust their mimicry, and that way they don't even have to hide that much anymore because they are literally invisible to the bee. So these are the sort of main results of the EARTH study. And I just want to tell you a little bit more about the EARTH project, which is the East Anglia Research Studentship. And the idea behind it was that beekeepers actually directly get involved in science by joining up with a research council, which is, in my case, it's the BBSRC, which is the Biotechnology and Biological Sciences Research Council. But they fund a lot of the biological research in this country. And the East Anglia beekeepers got together with the University of Sheffield and the Food and Environment Research Agency, so the National Bee Unit, to co-fund or apply for money through the BBSRC to then do this particular research project. And it was up to the beekeepers what the general topic was about, and then the scientists we said, okay, these and those, they suggested for the Varroa mite, and we said, well, these and those areas are still quite sort of murky, we still don't know a lot about, so it'd be sensible to write a research grant based on that, and then we send it off to my supervisor sent it off to the BBSRC, and they got funding together. But the big advantage is that the beekeepers could choose the topic, and also every year I spend about 2 to 3 weeks with beekeepers learning about the trade. So the first year I spent with hobby beekeepers, then the following year with commercial beekeepers and bee inspectors. And that way, as a scientist, you get a much better idea as to what the industry needs, and also which questions are sort of more pressing. And if I ever was involved in the sort of design of a treatment, I know what a treatment would have to look like to be sort of applicable to beekeepers and bee farmers. And the bee inspectors in particular gave me quite a good insight into the different diseases that are out there, which was quite scary. And I've seen some hives being burned, and it's not a nice experience, and it sort of motivates you to research these diseases further. And the big advantage was as well that I actually gave a lot of talks through different beekeeping associations, which means that I could not only update them on this project, but also because I go to a lot of conferences, I could give people insight into what's going on at the moment, and I could answer any questions that they had with regards to sort of current bee research projects that are going on. So I just want to finish by thanking my supervisors, Dr. Steve Martin and Roger Butlin, as well as Giles Butt from the National Bee Unit, our collaborators at the University of Kiel. They are proper chemists, which is always good because as a biologist, you always have to rely that you're getting your chemistry right. So Falco Driftwood and Sue Schamild made sure that I got all the chemistry bits right. I had my colleague Rian Gillam, who's working on ants mainly and how they recognise each other. And a big thank you to the BBSRC and obviously the East Anglia beekeepers for sponsoring the project and helping me also with the maintenance of my own hives, which has improved greatly since getting official lessons. So thank you very much for listening.
First of all, thank you very much for such an interesting presentation and explaining your method. Given that Varroa hops species onto the honeybee and it can adapt so well to different phases of the bee's life cycle, is it not surprising that Varroa has not hopped onto another insect species? Because I don't think I know of it anywhere else.
The Asian honeybee was obviously the original host, and even though the chemical profiles of the odour from the Asian honeybee is different to the European honeybee, there is, I think it's still similar enough for the mite to do it quickly enough to sort of get, hidden on the European honeybee, which is probably the reason why it wasn't that difficult for the mite to actually switch between an Asian to a European honeybee. However, when you have other insects such as wasps, for example, their odour is dramatically different. And also wasps don't have as many hairs as honeybees, and obviously the mite works with a lot of sort of hooking devices, and you need a hairy insect to do that. So that leaves wasps out. Wasps, as you all know, are naturally very aggressive. So our bees are actually quite calm, and I think that might be partly as well why they are quite accepting of Varroa mites. They're not I was just in Thailand earlier this year, and their bees are very different. So they're very, very aggressive, and it might explain also why they're a little bit better at sort of removing mites. However, the mite can be kept on bumblebees, but the problem with bumblebees is that they don't overwinter as a colony. And that is a problem to the Varroa mite, because it needs to have a host species that has a colony throughout the winter, because mites are very bad at staying alive when they haven't got the right humidity and the right temperature. And this is obviously all provided by a honeybee colony. And if they don't have that, especially during winter, that's it. So the honeybee is a very good host species for this particular parasite. And I couldn't think of any other insect that would fit these particular needs of the mite to be able to swap, because again, ants don't have a lot of hair, and also they don't interact that much with bees. So you would need an insect, that would interact a lot with bees, so you could get this sort of host switch occurring. Because if bees didn't interact as much with each other, there wouldn't be that much of a transfer between mites and bees, which is also why the bee trade obviously introduced the mites to the European honeybee from the Asian honeybee.
Thanks a lot for a super talk. Would you like to speculate on some ways of killing mites that might come out of your research?
Right.
In the most humane way possible, of course.
The interesting part for me as a biologist is that the mite itself doesn't produce a lot of compounds, because usually mite species also produce odours. And again, social parasites are very good at not producing their own odour, so that they can quickly adjust to the odour of their host. But we know that usually when an insect or a mite cannot produce a certain odour, that the genes for that are still there, so they're not lost. So you could look into, and I know obviously in America and in this country as well, at the minute they're looking at RNA interference, where you introduce and you change the genetics of the parasite, usually to destroy itself, but this could also be used to, if the genes are still there, to make the mite stand out chemically. So even if the method hasn't worked 100% with this sort of killing off the mite, at least it would stand out and the bees would be able to remove them. And obviously you could apply a powder, to that would, because mites and bees apparently vary in their natural sort of electrical charges. So you could, and there are powders out there that would just stick to one but not the other. So you could possibly impregnate them with an artificial odour, but all of that would need to be tested. But it's sort of down those lines where this research could be taken further.
Yes, I'm amazed by the intricacies of your research. I understand that the Varroa mite came from the Asian honeybee, where it had evolved, and they had a symbiotic relationship, but it's disastrous with the European. How come this?
To start with, in the Asian honeybee, the Varroa mite only goes into the drone brood, and they don't go into the worker brood. There is already quite a big difference there, which it could be because, that worker brood cells are quite small in Asian, and the mite needs to put its eggs somewhere, and the brood that moves around in the cell, and obviously any eggs that are not positioned properly are being squashed. And Asian honeybee worker cells are quite tight, much smaller than the European honeybees. And also the mite that, those mites that actually infect the drone cells, if the mite population gets too big inside the hive, the drones actually, because you get more mite families within one drone cell, they can't emerge. So the drone can't emerge because it's weakened so much. Because if you looked at the cap of a drone cell in the Asian honeybee, it is considerably thicker. It's really difficult to get off. You know, I collected some drone brood earlier this year, and it was quite a pain to get them out. You know, it's so thick. And if you have too many mites inside the colony, the drones can't emerge properly, which naturally keeps the mite population at a low level. And also because the Asian honeybee is slightly better at detecting them. But like I said, you know, they also swarm very quickly, Asian honeybees. And when you swarm, you naturally lose a lot of mites because the whole colony sort of goes out. And if mites drop during the flight, it's very difficult for them to get back onto the host. Whereas if bees mainly stay in the hive, like we have with our European honeybees, they mainly stay in the hive structure. The mites can still, even if they fall down, they can quickly move up and back onto a bee. So it's going to be a combination of those things.
What's the implication of your research for hygienic behaviour research? Yeah. If the bees actually can't detect the mite, they're not going to clean it off, are they?
And as far as I know, the hygienic bees are very good at detecting brood cells that have mites in them. And this could be because when the mite feeds on the pupa, it actually introduces infection as well. So there'll be a natural change in odour in that brood because of this. Because we know when you stress bees, their chemistry changes, so their odour changes. And that's also how the preliminary research, I'm not quite sure whether it was honeybees or bumblebees, shows that bees can tell sick individuals based on their odour as well, because that changes depending on whether they're healthy or ill. So it could be that hygienic bees are very good at picking out brood that are somehow, you know, different in their odour because they've been infested with mites, but it's going to be more to do with the pupa smelling differently rather than the mites, you know, the bees being able to detect the mites inside the cell. So it's got more to do with the cell smelling funny rather than the mite itself, because the main removal happens within, you know, removing sick brood out of the brood comb.